Like other scientists, Dr. Zavos had also read about Lorraine Young's findings.
We're going to begin doing human reproductive cloning as early as probably 30 to 60 days from now. Well, I do see that there's a problem, and unless you get...
For him, a problem found was as good as a problem solved. Horizon has tracked down a document in which Zavos claims he can actually make cloning safer by screening cloned embryos for the sort of errors Dr. Young found, and only using ones that look normal.
Frankly, I think the claims are ludicrous and absolutely irresponsible, and find there's absolutely no scientific basis for them. It's poorly referenced, and there's no real explanation of how he will do things. It just demonstrates a complete lack of scientific understanding, in my opinion.
Dr. Zavos couldn't be interviewed by Horizon because he's tied into a broadcasting contract elsewhere. But he claims that if the embryo has normal-looking methyl molecules on its growth genes, he will assume it's healthy. He and Dr. Antonori believe that tests like these will prevent many abnormalities.
We will check the complete procedure to monitoring to avoid 90% the abnormality.
But Dr. Young believes that the problem doesn't just affect the genes she's looked at. It could affect any gene.
There could easily still be problems in a whole range of other genes. He's going to be missing absolutely thousands of other problems. I mean, he's going to be missing everything.
In the last few months, Dr. Young's research has been backed up by other studies. Around the world, scientists have published the first results, showing that cloning sometimes causes vital methyl molecules to go awry on many genes. These results have led some scientists to a radical and devastating conclusion: that cloning can cause random errors in the switching of any one of 30,000 genes. The clone may develop normally until some key gene, like a time bomb, causes disaster. And they believe human cloning would be just as risky.
The most likely outcome would include late abortions, the birth of dead children, and perhaps worst of all, the birth of children which survived but which were abnormal.
And the risks don't end when we're born, because as our bodies change at key stages in our lives, we're dependent on genes switching on and off at the right time.
Things can go wrong at any time. There are some genes which, for example, are necessary to make a child go through adolescence and not really important before then. So if you have an abnormality in that gene, the child might not become a normal adolescent. And you could see failures at any time of development. That, I think, is what we should anticipate if anybody clones a person with our present techniques.
But there are other scientists who believe the case against cloning is hugely exaggerated, and that these early problems will be quickly overcome.
To take those very early experiments, which look at only a handful of genes, and try to make some grand statement that cloning will never be feasible in humans, or that it will be a very hard process, or to make any assumption of that sort, is unwarranted given the very preliminary nature of these studies.
Dr. Zavos and his team also think their opponents are exaggerating the risks.
Hello, Dolly.
He ridicules recent claims that Dolly herself may have something wrong with her brain.
Now, Dolly was doing okay until recently when we broke the news, and I was shocked.